Hello, welcome to Bio 204 Anatomy 2. This is Dr. Teresa, and in this video, we're going to go over the information for the circulatory system. The circulatory system consists of the heart, the vessels, and the blood, in contrast to the cardiovascular system, which is only the heart and the blood. The study of hematology is the study of blood. And I think we know this, that the functions of the circulatory system serve to transport nutrients to cells and tissues. It serves to protect the body by ways of inflammation and the immune system and through blood clotting. It also is one of the primary regulators of fluid. It helps stabilize the pH of the extracellular fluid, and it does have a lot to do with temperature control. Blood, because there's a large portion of it that has water in it, can transfer heat and conduct electricity. The components of blood, it is classified as a connective tissue because it has that extracellular matrix, which is the plasma. Within the plasma, you have water and proteins and electrolytes, etc. We'll go through those shortly. And then within that, you'll have formed elements mixed, and these will include blood cells and cell fragments. So in this picture, I think this is a helpful picture so you can identify what's what in a blood smear. In an entire blood smear, this first slide on your left, all of the little pinkish circles, those are your red blood cells. The purple dots that are floating around beside them, those are platelets. So you can see how different in size they actually are. And then the two really big cells on that slide, those are white blood cells. There are 7 kinds of formed elements. That includes all the types of leukocytes. So erythrocytes are formed elements within the blood, and these are your red blood cells. The platelets, I think we understand that these are fragments. They help with clotting. And they come from what's called a megakaryocyte. It's a special cell, and we'll get into that later. Leukocytes, these are your white blood cell group. There are 5 types. Of those 5, they are further broken down into 2 categories. So in the leukocyte category, you have granulocytes and agranulocytes. In the granulocytic category, a granulocyte on a stained slide will appear darker. They're generally purple, and they're going to hang out in the cytoplasm of the cell. And they are really like little packets of chemicals to help the cell fight pathogens, invaders, and toxins. In an agranulocyte, these guys don't have granules. Their job is different. So the components of blood, again, this picture is nice just so you can get a size differential. You can compare that RBC, or the erythrocyte, compare it a little bit more intimately to the platelets, and then to the different types of white blood cells. When you take a blood draw, within that whole blood sampling, you put it in a tube or into a capillary tube, and then you put it in a centrifuge. What the centrifuging does is it will spin the heavier elements to the bottom of the tube. It separates everything. So if you look at the picture on this page, you can see the top layer is what we call plasma, and this makes up 55% of whole blood. Then you see a little layer called the buffy coat. It is part of the formed element category, but it's less than 1% of your blood. This is your white blood cells. And then the erythrocyte category is going to be the heaviest category of formed elements. So it'll be on the bottom of the tube. And this is going to make up approximately 45% of the whole blood sampling. Now, within that plasma, it does, again, this is the matrix of blood that makes it connective tissue. It has water and proteins, nutrients, electrolytes, nitrogenous waste, hormones, and gases. The difference between serum and plasma is that serum won't have fibrinogen. Fibrinogen is going to be involved in blood clotting. Within the plasma, we will see three primary categories of proteins. They will be albumins. Albumins are the smallest protein in the plasma. They're the most abundant, and they will be formed in the liver. The globular proteins, or globulins, these are antibodies. So antibodies will float around in the plasma. They provide, and I'm fairly certain we're all aware of this, immune system functions, and they're formed by the B lymphocytes. And then there's the fibrinogen that we mentioned. This is a protein that is formed in the liver, but will circulate within the plasma and be ready to do any work on blood clotting that may be required. Within the plasma, we are going to find nitrogenous compounds. This is from the breakdown of amino acids, and this can be from amino acids in your diet, or if your body needs amino acids to do some work, it will break down your own tissue. Nitrogenous waste can be in the form of a compound called urea. And we can see this in a blood draw measure to determine the function of the kidneys. Nitrogenous waste, or urea, that's an end product of catabolism or the breakdown. We're going to find glucose, or sugar, within the plasma. Vitamins, fats, cholesterols, phospholipids, and minerals. We'll find our oxygen and CO2 values along with nitrogen itself. This is going to give us an indicator of how well that transfer process at the cellular and tissue level is going in regards to blood carrying oxygen and blood taking away waste product. Electrolytes then will make up a portion of the plasma, the primary one being sodium. Again, I'll put a little cheerleading plug in here for sodium. It is not the devil. It does pretty much everything in your body, and we require it. It just can't be overloaded. Other systems have to be in play in order to regulate it. When we talked about viscosity, viscosity is the thickness, really. It's gonna, and something that's thick, it's gonna be harder to push through your vessels. So think of your vessels as like a tube. If the fluid in the tube is like mud or tar, the flow will be different than that of something that's not so viscous or a little bit thinner in composition. When we talk osmolarity, if you remember, has to do with particle size and osmosis. The total molarity of those dissolved particles that cannot pass through the blood vessel wall. So they're too big, or for some reason they can't go through the processes of diffusion. So osmosis comes in here. If the osmolarity of the blood is too high, meaning the concentration of particles is too high within the system of vessels, it's going to absorb water into the vessel passageway, increasing the blood volume. When we increase blood volume, we increase blood pressure. If the osmolarity is too low, the opposite thing happens. So we have high osmolarity or concentration of bigger particles that can't make it through the wall. In the first example, if we have a low concentration of that within our vessels, the water within the vascular system will leave, and then it will go into the tissues. So we'll have a drop in blood volume, which will equal a drop in blood pressure, and that's going to give us tissue edema. Hypoproteinemia, this is a deficiency of the plasma proteins that we just went through. We can see this in extreme cases of starvation, but primarily we're going to see that in liver and kidney disease, also severe burns. Then there's a condition called kwashiorkor. This is in children with severe protein deficiencies because they were fed on cereals too quickly. So they're going to have thinner arms and legs and a swollen abdomen. The way the blood is formed is really cool. So let me talk through some of the language. We have the pluripotent stem cell, or the PPSC. This is going to be the start of all of your formed elements. So yes, red blood cells, but also it's going to form the platelets as well as the white blood cells. The colony forming unit, this will be the first committed cell in the line of differentiation, where we have a definite direction of what that cell is going to be when it wants to grow up. From the colony forming units, it's going to break into one of two paths. Myeloid hemopoiesis, which is carried out within the bone marrow, or lymphoid hemopoiesis, which is carried out within lymphatic organs. So we'll see a picture of that in a minute, and that will make more sense on how they break down and what they form. Here's an example of how a red blood cell gets produced. It starts with the PPSC, gets directed by some stimulus to go into what is called a CFU, colony forming unit. It then forms what's called an erythroblast. The erythroblast will then form into the reticulocyte, and only about 1.5% of all red blood cells floating around that are measurable are reticulocyte. And the rest of them get formed into the mature erythrocyte. So the lifespan of that mature erythrocyte is 120 days. The development takes 3 to 5 days to form this from that stem cell to the mature cell. This is the picture I was talking about. This is a great picture. We'll see it multiple times. You can see within the section of the femur that's represented here, that's representing, it's starting in the red bone marrow here as the stem cell. It will get direction then to form into either the myeloid stem cell or the lymphatic stem cell line. In the myeloid stem cell line, that is going to produce your erythrocytes, platelets, basophils, eosinophils, and neutrophils. So all of your granulocytes get developed here or from here. It also will make your monocytes, which is in the agranulocyte category. When we look at the lymphatic stem cells, the ones that will get produced within lymphatic tissue, those are going to be your B lymphocytes and your T lymphocytes. Now, the B lymphocytes, which are agranulocytes, these will get processed in the bone marrow. Their products get shipped out. The T lymphocyte, these cells will get processed within the thymus. T for thymus, B for bone marrow. Remember that red blood cells are a biconcave disc, meaning that they have a thinner section in the center. And this serves a purpose as far as increased surface area and therefore increased carrying capacity, oxygen and other gases. Now that the gift of that small central space within that biconcavity, that also will serve as a point of faster diffusion because it's a thinner space. We already have touched on this, but the two principal functions of an erythrocyte are to carry oxygen from the lungs to the cell, to pick up carbon dioxide from the tissues and bring it to the lungs. And this has to be a system that functions highly, because any lack of oxygen, even if only for a few minutes, will we're going to have necrosis or cell and tissue death. So again, let's talk about, or just review what I just said, the form and function portion of the red blood cell. These guys will lose nearly all of their organelles during development. They lack mitochondria and they lack a nucleus, so they're not going to go through cell replication. Let me say that again. They lack a nucleus and DNA, because potentially, if it were a different kind of cell, it could still have DNA but no nucleus. But in this case, since they're cells within our body and they are eukaryotic, it does not have a nucleus. Therefore, no protein synthesis and no mitosis. They just keep getting made, okay? Now, the fact that they don't have mitochondria is relevant. The form of energy production that they have to go through is going to be primarily, they'll have to do some glycolysis and then some lactic acid or some type of anaerobic fermentation, likely lactic acid. So when we talk about gas transport through this cell, I already mentioned the increased surface area. Now, it's also relevant to know that 33% of the cytoplasm is hemoglobin. I think we're aware that hemoglobin is the carrier molecule. And we will see a picture of that shortly, and we'll describe that further. Also, what we have in the cytoplasm, really important molecule, is carbonic anhydrase, or CAH. Remember, the blood is great at buffering things. So the carbonic acid molecule is, if you remember from like chapter 2, AP1, the carbonic acid molecule was used to perform buffering, both in the lungs and in the kidneys, and this is going to be relevant within the blood itself as well. Remember, we have to be able to balance and manage a pH or things start to go wrong. So here is our hemoglobin molecule. Remember, globin chains are tertiary structure proteins. A quaternary structure protein, which is a molecule of hemoglobin, has two or more globin or globular proteins bound together. And within hemoglobin, there's also a central group called the heme group. The heme group will have the center of it being iron, ferrous. Now, you'll notice on this picture as well that on this picture, we have it labeled as beta alpha. This would be in the hemoglobin in an adult due to these linkages, and all these refer to are molecular linkages. We're going to just briefly touch on the fact that this will be different within the fetus as well as the child. Remember that globular proteins, again, that's a tertiary structure proteins, kind of a wound up ball of yarn. So the difference in how these guys link are going to give it its function. As we learned way back in the chemistry section, is that the structure of a protein is super important. If it starts to denature or unwind or become what it's not, it will not function right. So here's the differentiation between adult hemoglobin, which has 2 alpha and 2 beta chains. That's the picture we saw in the previous slide. In a child, you have 2 alpha and 2 delta chains. Again, it's just a linkage point. And then in the fetus, you have 2 alpha and 2 beta chains. When we are looking at a whole blood sampling, the concentration of hemoglobin is relevant to health. Therefore, the concentration of red blood cells is also relevant to health. So we want to look at these values within the healthcare setting to see what's happening metabolically. Hematocrit is a measurement of the packed cell volume. It is a percentage of the whole blood volume, which is composed of the red blood cells. If you remember that picture on that slide previously of the test tube that had been spun through the centrifuge, and it has the red blood cells on the bottom, the plasma on the top. We're looking at the red blood cells on the bottom. Men will generally make more than women in all of these instances. Again, if we break that down, any difference between male and female values, numbers, productions has to do with hormones on board. Hematocrit will also vary with the state of hydration. Then we want to look at the hemoglobin concentration of whole blood. Again, men have a higher level of hemoglobin in general, meaning that they're going to produce, or their oxygen carrying capacity is going to be higher than in a woman. When we actually want to do an RBC count, also men are making more. Now let's go back to this picture where we're talking about erythrocyte production. Again, we started with the pluripotent stem cell. It goes into what's called an erythrocyte CFU. That's the first committed cell in the line that knows what direction it's taking. This is going to be inspired by a substance called erythropoietin, or EPO, that comes from the kidneys. So it is a hormone that's going to impact what gets made. Erythroblasts then will multiply and synthesize hemoglobin. If the nucleus gets discarded within this process, this is where the reticulocyte gets formed, and it's called that because of its endoplasmic reticulum. We're going to see more of these if we have blood loss or COPD. Iron metabolism. This is a huge deal. Now, it says low absorption rate. Okay, I would say that that is highly dependent on the chemical formula that iron comes in. Not all of it's going to be the same. And something that is more, I'm going to use the word natural, but I don't have another word for that. And it's, we'll say more complete, that's better. More complete, dietary wise, it's going to be easier for most people to break down and absorb. That does, however, depend on the digestive system. So we're understanding through iron metabolism that there's a large portion lost through iron, feces, and then through bleeding. Now, in this category, men lose less and women gain more, and that makes sense due to the menstrual cycle. We just talked through this, the construct of hemoglobin, so we understand that iron's at the center. So this is how it works, that we will take in dietary loads of ferric form iron, Fe3, as well as ferrous form iron, Fe2. Anything that's in Fe3 form will need to be converted into Fe2, and that will get done by way of being broken down in the stomach by the stomach acids. There is then a substance called gastroferritin, which will bind to this ferrous form of iron. It will take it to the small intestine, where it's absorbed into the blood. In the blood, it's going to bind to a substance called transferrin. This is to transport it to the bone marrow and the liver. Once it gets to either bone, muscle, wherever it's going, okay, this is where the hemoglobin process begins to happen. So we call bone marrow hemoglobin. When it's in the muscle, it's myoglobin. And all cells will use cytochrome. We'll use this for cytochromes in their mitochondria. So there's a high relevance of this pathway and system functioning very well. In the liver, there's a substance called apoferritin, which will bind to create then ferritin for storage. Now this picture, I'm not going to read this picture. I'm going to let you look at it because it walks through what I just spoke about. Takes the words and puts it into pictures. So the formation of red blood cells or erythrocyte hemostasis, it's a negative feedback loop. We learned about feedback loops like in chapter 1. Remember, negative feedback loop will get the information back about how much is circulating within the body or whatever, and it will shut the process down. Whereas a positive feedback loop will receive the stimulus information and keep the process going. So within this negative feedback loop, there would have to be a detection of decreased red blood cells, and we call this hypoxemia. So what would stimulate this or give the message to go into this process? If the oxygen levels are too low, okay, partial pressure of oxygen, that can be from the person's inability to breathe due to restricted airways, or high altitude, or intense exercise. The kidneys will get the signal to stimulate the release of that erythropoietin that we mentioned a few slides up. And it will stimulate the bone marrow to begin the process. Remember, this process of forming a deficit or correcting a deficit in red blood cells takes 3 to 5 days. So we've talked multiple times about the body's ability to recycle. So this is no different. And actually this recycling process, we have everyday evidence of this in our urine as well as our bowel movements. So what happens is the RBC will get to the spleen, okay, which is the graveyard. It will be lysed or, burst, explode, break itself down, become broken down by macrophages in the spleen. What happens is they will digest the membranes, and then in regards to the hemoglobin itself, they're gonna hydrolyze the hemoglobin into its globin groups and the heme group. From there, the iron will be removed. When the iron is removed, we have a pigment that's converted to something called biliverdin, which is green. Biliverdin is then converted to bilirubin, which is yellow. This will get released into the blood plasma and will make its way to the kidneys. And this is part of the reason our urine formation and filtration process ends up with yellow urine due to the presence of bilirubin. The liver then will remove bilirubin from circulation and secrete it into the bile. The bile is gonna end up in the gallbladder, and then the gallbladder will release this into the first part of the small intestine. This compound will get broken down further by the flora in our gut and deliver us our general brown feces. As we know, the color can vary. It'll be dependent on what you do eat. The ability of all these mechanisms to work well, and if there is a blockage within the ducts of the gallbladder, common bile ducts, there's a pancreas issue, you may see a change in the color of your stool. When we talk about jaundice, oh, by the way, that previous slide is just what I walk through. There's pictures for you to understand it. If you like the pictures, set to words. I won't read that for you. Jaundice, we know, is yellowing of the skin. We think that it happens most commonly within children, but if a person has liver damage, they're also going to experience jaundice. What happens is there's a rapid hemolysis. So within all of these processes within the body, all of physiology, there's a requirement for time, for course correction, so that the body can be balanced a little bit better. If you have a blocked bile duct, I mentioned this as far as a change in color in the stool, but also this can contribute to jaundice as well, because it will lead to the bilirubin being concentrated more in the blood. You might get yellow skin, a yellowish tint to your skin, and the sclera of your eyes will be yellow. Newborns may have this just because their liver is not fully functional yet. Vitamin K is not even secreted by bacteria yet when they're born. And the way that this is corrected is through UV light, which is super cool. If we apply UV light, sunlight to the skin, that is going to help break down and convert the bilirubin to a form that the body can actually eliminate better. Let's talk through the disorders that erythrocytes might experience. Polycythemia is the term we give for an excess of red blood cells in the blood. Now remember, we expect it ideally to be in a certain volume, have certain range of numbers of RBCs, and a percentage of a whole blood trial. So when any of these values become aberrant, we could potentially have physiological issues, one being polycythemia. And we will measure that via our comprehensive metabolic panel. Okay, we'll look at what the relationship to red blood cells is to everything else. So primary polycythemia, or polycythemia vera, is what we call something that is cancer of erythropoietic cell line and red bone marrow. You may have an RBC count as high as 11 million with a hematocrit at about 80. Remember, hematocrit is supposed to be around 45% of the whole blood sampling. So it doubles in polycythemia. In secondary polycythemia, this can be from dehydration or respiratory failures, diseases, high altitude, or intense exercise. And you can get your RBC count up to 8 million. The dangers are this. Because there are more formed elements, packed cells, right? This will increase the viscosity and make it thicker, which is going to then drag water into the vascular system. So we're going to increase the blood volume, thereby the pressure. So disorders like this, if we have aberrant erythrocyte production going crazy somewhere, can lead to embolism, stroke, or heart failure. Now, on the other side of the spectrum, anemia. We have 3 different types of anemia that we'll talk about. Anemia is we have an inadequate number of red blood cells or hemoglobin synthesis. This can stem from kidney failure or the production of the EPO or the erythropoietin gets interfered with, thereby not being able to stimulate the process. It can come from iron deficiency. We all know that one. It can come from an inadequate B12 measure. If you have low B12, this can come from not getting it in your diet, not breaking it down. I'm going to add that one in there. Or the lack of what's called intrinsic factor. This is relevant because B12 is needed for the synthesis of these guys, DNA synthesis specifically, but it's not going to be able to help formulate these cells. Hemorrhagic anemias, that's from trauma, or you have a heavy menstrual cycle and you can become anemic. Hemolytic anemias stem from RBC destruction. This can happen from drugs, penicillin, poison, or a malarial infection. Now, the danger of an anemia is it could create tissue hypoxia and necrosis, meaning you're going to get lethargic, you don't have any energy, shortness of breath upon exertion, life-threatening necrosis of brain, heart, or kidney. If you're not carrying oxygen, okay, because you have some type of anemia, which is going to impact your oxygen carrying capacity, you can hurt yourself. The blood osmolarity will be reduced in an anemia. Remember, if we have a lower osmolarity in the blood because there's not as much there, that's going to take water out of the vascular system and push it into the tissues. So we're going to have a drop in blood pressure and tissue edema. And if the viscosity is low, same thing happens. When we have sickle cell anemia has it's a genetic aberration of the shape of the red blood cells. It's usually in people of African descent, where the hemoglobin does not bind oxygen well. The shape of the cell makes it rigid and sticky, and they can clump together and block vessels. So this can cause a lot of pain, but this can also cause or lead to really anything that would be like polycythemic in nature. Okay, let's talk through blood types. Blood typing is relevant because it gives us sort of a peek into a person's biochemistry, really. This is a genetic trait, and A blood types are dominant, B blood types are dominant. AB is a co-dominant trait, if you remember all of that from your Bio 1 stuff, how they will mix and match. And O blood type is a recessive allele. So we're going to go through the understanding of what makes a blood type. And this has to do with antigen antibodies. Remember, antibodies are globular proteins. So they have a particular shape and they're relevant and they float in the plasma. We just went through that. So the relevance of antibodies floating in the plasma has a lot to do with the antigen, the cell surface marker on your RBCs. Now, if you remember back in AMP1, where we walked through the parts of the cell membrane, there's a particular one there called a glycoprotein. It's a protein that's embedded in a cell membrane and has like a little carbohydrate flag on it. This will form what's called the glycocalyx in general, but also when we're talking about red blood cells, it's going to form the antigen on the surface of the cell. in order for the body to determine what is self and what is other. This is relevant because when we go to get or give blood transfusions or organ transplants, if these patterns are not compatible, we're going to have a reaction and someone could die that way, and we don't want to do that. And it's not super straightforward. Okay, there are elements at play when a person gets a blood transfusion from another human. Yes, their blood typing may be the same, and we would think that everything's going to go well. That is true. I hope you're learning that that's textbook, and it doesn't always work that way. There are factors beyond that we don't know about yet. I will keep telling you we don't know everything about the human body. Okay, we put a lot into practical application without a full story. We do wonderful magical things. This is the case of that. Okay, so there is great care, even beyond knowing that the blood types match, that you have to monitor people receiving anything like this. So that's a process called agglutination, or clumping, when we have an antibody antigen response happening within the body due to the presence of something foreign. That's not going to work. Agglutination is when the antibody, that's the part floating around in the plasma, will bind to the antigens on the cell surface. It's attacking it. So it will be in opposition. For example, and I'll go through this in varying ways and multiple times. If on the surface of a blood cell you have A antigens, that makes your blood type A. Now in the plasma floating around, you cannot have anti-A antibodies, or it will attack you. It will attack those red blood cells and you'll die. So it will be antibodies of opposition, meaning this: you can have A antigens on the surface, that would be your blood type, and you would have to have B antibodies floating around in order for there not to be an immune response against the blood transfusion. So here's a good picture. This is a very nice picture. And we went over this in lab. In your activities, you learned a little bit about the blood types. So again, what is on the surface of the cell is the antigen, and then the antibodies are what's floating around and will be the determiner of self or other. As I mentioned, if you have A type blood, you have A antigens, B would be B antigens, and then we have to put into play the Rh antigen, positive or negative. Now, in an O blood type, you're not going to have any antigens on the surface except maybe the Rh factor. So O blood types have neither A nor B antigens on their surface, but they may have the Rh antigen or Rh factor. In an AB blood type, you have both. Okay, our reference point is the actual cell. So what is on the surface of the cell will deliver you the blood type. If doing a blood typing exercise, you apply an antibody or anti-serum that creates agglutination, that is the opposite of what you want, and will tell you or deliver you evidence of what antigen is on the surface. It's worthy to note that a type O person is the most common type of blood, and the rarest is AB. Again, AB is a codominant trait where both of those are dominant blood types within humans. So if you can kind of put that in your head, remember those Punnett squares. We're not doing that in this class. It's just for awareness. Your blood plasma, again, has the antibodies, or what we would call an agglutinin. The antigens, by the way, another name for them, agglutinogen. I already went through agglutination. It is when they clump, when we have an antigen antibody response, because there is a particular antigen on the cell surface, and then you add antibodies or have antibodies. We can put it in the context of you getting a blood transfusion. Pretend you have A blood. That means you're going to have anti-B antibodies floating in your plasma. So if somebody messes up and they give you B blood, the B antibodies are going to attack B blood. This is dangerous because that agglutination, sort of like a sickle cell thing, right? It clumps and it's going to clog vessels. We're going to shut down vascular flow. And then cells and tissues will not get oxygen and required nutrients or be able to do the exchanges. When we talk about, we've all heard universal donor, universal recipient. Universal donor, that can go to anyone with any blood type. Good thing it's the most common blood type. Remember, the O blood type has no antigens on its surface. So that O is a 0, okay? That if you have 0 antigens on the surface of your red blood cells, you're an O blood type. Now, this is valuable because if you have to guess, you can give someone O blood type without having an antibody response, because the O blood type, or the red blood cell with no antigens on it, it's not going to have or respond to anything of an attack. So if you get transfused with an O blood type, but you're an A, you're safe, because you would have anti-B antibodies floating around in the plasma. Because there's no antigen on that O, you would not see or high prevalence, I should say, of an agglutination response. Now, the universal recipient, the reverse of that, is the AB. Remember, AB has both antigens on the surface of the cell. So if you're an AB blood type, what you have floating in your body are AB, you will not have antibodies to either. So you can receive A or B or O, okay, because there will be no antibody response against your red blood cells. Here's a nice picture of that. If the words didn't make sense, I won't read through this again, but pretty much this is what I was saying. So these pictures are nice. If these make sense to you and help you as far as understanding the blood typing, that's important. Now in this slide here, this is going to be relevant for you to understand that in the top response, we have no clumping, no agglutination, meaning no attack of an antibody that's floating in the plasma onto the cell. If, however, you choose the wrong blood type, or, again, this isn't as simple as we want it to be, we would like it to be that cut and dry, it's not. Okay, sometimes even with that, things are gonna go wrong. Now let's talk about the RH group. The RH group, well, that's our positive or negative identifier, and it's another antigen on the cell surface. There are really 3 of them, C, D, and E, D being the most reactive. So that's the one we talk about. So if if RHD is present, the patient is considered to have RH positive plus, it's on there. Okay, so if you have RHD, you're a positive blood type. Now, if you have RH positive, if you're positive for the RH factor, meaning this antigen is on your cell surface, then you cannot have antibodies against it floating around. If that happens, we will have problems. So this is yet another potential hazard when we're trying to understand blood transfusion and organ transplants also, where this guy comes into play, and we touched on this. If a pregnant mother, they find that the first pregnancy, the mother has an opposing RH factor, to the fetus, it will require some intervention for the second pregnancy. And that is so that if a woman is Rh negative, okay, no antibodies for the Rh factor on her cells, and yet the fetus is developed genetically to have Rh positive, meaning this antigen present, there can be an attack across the placenta of the fetus, and this can cause problems for the baby. And this will require the Rovium shot. It will bind the agglutinogens or the fetal antibodies in her blood, so she will not form antibodies against the blood of the fetus. If it occurs, the child can have anemia or like a toxic brain syndrome. Okay, let's dive into leukocytes. Leukocytes, that word should tell us we are dipping into white blood cells. Remember, the white blood cells make up 5 of the 7 formed elements of your whole blood sample. There are 2 categories within the leukocyte category. They are granulocytes and agranulocytes. Granulocytes are your neutrophils, eosinophils, and basophils. Your agranulocytes are your lymphocytes and monocytes. We're going to get into that. I'm going to break it down why they would be present and what you would see. So these are the least abundant formed element. We don't want to see these in a high concentration, because when we do, that tells us we're off to war, our immune system is battling something that needs controlled, okay, for our health and wellness. Now, can you have temporary rises? Absolutely. So one thing that I don't like to do is get freaked out when we have a blood draw that shows us out of range limits. I think you need to do some pattern work, meaning do multiple draws within multiple weeks. Okay, in the interim, again, one thing that conventional medicine champions at is delivering us time. We control an acute situation through pharmaceuticals and diagnostic testing to give us an idea of where we are. And then we do the back end work as far as lifestyle changes and nutrients in order to nourish, replenish, and repair anything that may be askew. Can this change? Yes, I've seen it. I've done it to myself. Okay, so just because today you get diagnosed with something doesn't mean that diagnosis sticks. And I would encourage you to, if you're delivered ever a contrasting health situation, you go look for solutions. If you can't find what you're looking for in one box, you go look in another one. The function, we all know this, white blood cells, they protect against infectious microorganisms and other pathogens. And the way that you're going to be able to identify them, and you will have to, it's either on a homework or a quiz or something. I think the pictures are black and white though, but you should be able to determine what type of leukocyte you're looking at by the granules, by the size and the shape of the nucleus. So really, they only spend a few hours when they're distributed into the bloodstream, and then they migrate to where they're supposed to go. They do retain their organelles so that they can perform protein synthesis. So this contrasts the red blood cell. So granules, I keep mentioning the granules within a leukocyte. Granules are chemical warfare. They're little chemical packets of stuff that a particular cell, when it determines something is not supposed to be there, it's when we get into the lymphatic system, you're going to see this strongly. It's amazing. It's like we have a militia in our body that seriously use like military tactics, or that's what it seems like. So these granules help break down, digest, and recycle foreign invaders and the components of them. So let's break them down in multiple ways. So in the granulocyte category, you're the most abundant leukocyte collectively in both granulocyte and agranulocyte categories is the neutrophil. That's the one that we are going to see the most of. If we look at blood or blood smear, we would expect to see those. These guys are going to fight or be stronger in bacterial infections. Eosinophils, these are going to stain differently, okay? With the stained eosin, they will be more pink, and their nucleus will be bilobed in the eosinophil. The eosinophil is going to be present also if you're fighting a parasitic infection or an alert. If you have an allergic response happening, they're going to be higher. Maybe collagen disorders as well, they would show up. Central nervous system issues. But if you look at the concentration of them, they should not be strongly concentrated within that Buffy code, which is what you would look at. Okay, this is the differential part where you break it down and compare them. Now, I didn't mention, but the neutrophils have a 3 to 5 lobed nucleus. And we're going to see pictures of this, so it'll be helpful when you see the picture. The basophil, you should have right around 1%. These guys are going to be present in histamine reactions, as well as within the production of heparin for blood clotting. So we have a histamine release to help with fighting things that should not be there. Okay, they have large violet granules, okay, where you pretty much can't see the nucleus of this guy. In the agranulocytic category, you have lymphocytes. They are collectively the second most abundant WBC in a whole blood sampling. Their nucleus is going to take up almost the entire center of the cell. There's almost no cytoplasm seen, and you will not see granules either. And the monocytes, these are the biggest white blood cell, and they're going to have a horseshoe-shaped nucleus. So if you were trying to differentiate here, I think the lymphocytes are easy to see. Again, that nucleus takes up almost the entire cell when you're looking at a stained slide or even a black and white one. Eosinophils will be stained kind of pink, and they'll have a bilobed nucleus. Now, I think when we're looking at the monocyte, just recognizing they are going to be the biggest thing you're looking at, and they will have that horseshoe of a shape nucleus. The neutrophil, you're going to be able to determine that with the connected lobes of the nucleus. And then the eosins, again, if you're looking at color slide, they're going to be more pink. I already mentioned this part, but I'll walk through it briefly again. We're going to see neutrophils in bacterial infections increased within bacterial infections because they will phagocytose. So they're going to release those antimicrobial chemicals, those granulocytes. Eosinophils, again, we'll see them in parasitic infections, allergies, and some other disorders. They are also phagocytic. These guys are going to form an antigen antibody complex. So similar, okay, in activity as we saw within agglutination reaction in the red blood cells. We're going to have an antigen that responds with an antibody floating around. These guys are like snipers, and they release enzymes that will chemically dissolve what they're trying to dissolve, what's in their way. Now, the purpose of the basophil, we're going to see this an increased number in chickenpox. It's viral. Sinusitis, that's who knows what the origin of the pathogen is there. That could be viral, bacterial, or parasitic, actually. Okay, and you're not going to have one type on board doing the work. Usually, you're going to see multiple. And then diabetes, which is interesting, okay, because if you understand that diabetes, it's rooted in inflammation. Inflammation is a process of the immune system. So when we have systemic inflammatory responses, we're going to see basophils more active in their secretion of histamine, which is a vasodilate, and heparin, which is an anticoagulator. Okay, so here this will help us identify our granulocytes. From left to right, we see neutrophils, 3 to 5 lobe nucleus. We can see it stains purple. And, you know, definitely they're all bigger than the RBCs, which are all the little pink guys in the background. Now that eosinophil, again, stains pinker, and we can see its bilobed nucleus attached in the center. The basophil, the granules obscure that nucleus. When we get into agranulocytes, these are my favorite ones to talk about. I think they're relevant, particularly right now, but always. Okay, again, these are the second biggest category of leukocyte within your body. We're going to see increased number in different types of infections and responses, but very common in viral infections and cancer. What they will do is go through a process of what's called antigen presenting. So it is an antigen presenting cell, meaning it's going to lock on to something that it doesn't know what it is, and it's going to take it to the judge and the jury for sentencing to see what's supposed to happen. Lymphocytes will coordinate actions of other immune cells. They will secrete the antibodies, and they are part of the reason our immune system has memory. We have B lymphocytes, which we mentioned. They are kind of processed in the bone marrow, B for bone marrow. T lymphocytes are processed in the thymus gland, T for thymus, and then the natural killer cells. When we get to the monocyte category, I mentioned multiple times, these are the biggest ones. This is the third most numerous category of leukocytes across the board, and they will be our macrophages. So they'll leave the bloodstream and go do what they need to do, looking for debris. So we see this in the central nervous system. We see this in the integument. Also has the activity of that antigen presenting process. Here on these two slides, relevant to take a peek so that you can understand the difference between a lymphocyte and a monocyte. Dark purple staining of the nucleus, but that lymphocyte, that nucleus takes up the whole entire center of the cell. In leukopoiesis, we went through erythropoiesis previously. Remember that they all start from the pluripotent stem cell, then they get differentiated. Within the leukocyte production or WBC production, they will go into the myeloblast category, which will produce neutrophils, eosinophils, and basophils. And we're going to get to see that picture again. So the picture will make these words a little bit easier to understand. The monoblasts then will form monocytes, and then the lymphoblasts will give rise to all of the lymphocytes. The T lymphocytes will complete their development in the thymus. So they're still going to start in the bone marrow, as we are going to see that map again. The red bone marrow is also responsible for storing and releasing granulocytes and monocytes. So here's that picture again. So everything I just said on that last slide, here we go, okay? So here is our pluripotent stem cell that begins in the red bone marrow. Now that's gonna break apart. Here are our lymphoblasts, which will give rise to B lymphocytes, which are processed within the bone marrow. And then our T lymphocytes, which will get sent off to lymphatic tissue, thymus, okay, for maturation. When we were talking about the monoblasts, okay, this comes from a different stem cell deviant. The monoblast will produce eventually that agranulocyte called a monocyte. Here's another picture saying the same thing. As they start from that pluripotent stem cell, they will be directed and committed to what they're going to be when they grow up. When they get to the colony forming unit via some type of stimulus, is some type of feedback loop, is making the body know which direction to push something, if that makes sense. Okay, there will be some type of biochemical event that's going to aware or make aware these stem cells in the bone marrow to do a particular type of production. And that could be anything. Okay, if you come into contact with carcinogens, that could inherently drive this line in a direction of confusion in the body, and it will go wrong. Now, we mentioned natural killer cells once. When we get to the lymphatic chapter, that's going to be a little bit more apparent. But as we look at our mature cells and where they came from. Down at the very bottom, we have that natural killer cell, which is going to be a break off of the lymphoblasts. So as we're producing the lymphocytes, we're going to have natural killer production as well. It's good to note that white blood cells do not stay in the bloodstream. Granulocytes leave in 8 hours and live 5 days longer than an agranulocyte. The monocytes leave in 20 hours. They will transform into their macrophages, and then they can hang out for years. So that's really relevant. Now, the lymphocyte category is what we think of when we think of long-term immunity that can last decades. This is where that cellular memory. Now, I want you to think about that in context. They don't have quote unquote brains, right? It's not that kind of memory, but it's fascinating. Our body goes through these cyclical patterns that we are not consciously directing, and yet something is stimulating them to either go right or go wrong. Now, when we get into the disorders of leukocytes, leukopenia, this is a low WBC count, below 5,000. This can be caused by radiation treatments, poison, infectious disease. And then if we drop our leukocytes collectively down, we are at risk for infection. Leukocytosis, this is when we have high white blood cell count, anything above 10,000. This can be caused from infection, allergy, disease. The differential counts, if you get a CBC and diff, okay, identifies the percentage of the total WBC count. It breaks it down into each cell type is what the differential does. Okay, it's not an absolute count. So, for example, if you get that done, you might have, 10 basophils, but compared to the rest of the cells that were measured in that blood draw, that's going to give you the differential count in contrast to the total white blood cell volume in a sampling. Leukemia is a cancer of hemopoietic tissue. So when we have leukemia, we're going to have high leukocytes and their precursors. So if you do a CBC and diff, you're going to see high, WBCs collectively out of range, and then break that down. And you can kind of get a clue. I love looking at a CBC, FYI, because I think it just gives us a ton of information on the direction of what the body is working with now. Myeloid leukemia would be, remember, from that myeloid portion of that stem cell line, and controlled granulocyte production. So we have something up primarily in that red bone marrow and beyond, really. If we have a lymphoid leukemia, we're going to have higher levels of our lymphocytes being monocytes, okay, or lymphocytes. Did I say it, monocytes or lymphocytes? An acute leukemia may appear really quickly and progresses rapidly, and this one would be one that's hard to catch. If you have a chronic leukemia, it can go undetected, like you wouldn't have any symptom or awareness that it's happening, which is, why we should pay attention to our body and its symptoms and its signals, and also go periodically to get yourself evaluated in some way so that you know where you're at in case something starts to happen. You can catch it maybe a little bit faster and have a better prognosis. So this slide is interesting. It does compare normal blood, top slide, and then leukemic blood. So we have, a really high percentage of white blood cells where they should not be. Now we are going to get into the function of platelet formation and blood clotting. Remember that platelets, these are fragments of cells. They are fragments of the megakaryocyte cell, which we saw within the stem cell line. A normal platelet count will be somewhere between 130,000 to 400,000, which is a big range. They function to secrete vasoconstrictors that will help reduce blood loss. I think we know that as well. If you don't know what a vasoconstrictor is, it's some type of chemical or stimulus that is going to change the diameter of a vessel. They will stick together to form something called a platelet plug. And this is so that within a vessel or tissue, it can seal up small breaks. It will secrete something called a procoagulant, which is a clotting factor to prevent clotting. This will initiate the formation of clot dissolving enzymes. When this happens, they start attracting neutrophils and monocytes to the site of inflammation. And remember, neutrophils are generally for bacterial infections. However, they act as, phagocytes. They will secrete growth factors that will stimulate mitosis to repair the blood vessels as well. Regarding platelet production, it goes through a process called thrombopoiesis. I don't think that should surprise us at this point. We've gone through erythropoiesis and leukopoiesis. Now on to the next one. Again, a stem cell that starts out, same cell line, pluripotent stem cell, develops into a megakaryoblast. The megakaryoblast repeatedly replicate DNA, but they don't divide. They form big cells, hence their name, with a multi-lobed nucleus. The megalokaryocyte then develops from the blast. It will live in bone marrow adjacent to blood sinusoids. So, pathways to deliver this. This is the cool part. These megakaryocytes then will have long tendrils or like fingers of cytoplasm that will protrude into these sinusoids or flow cavities. As the blood flows, it will drag with it parts of the tendril or the roots. It's like it has roots. Here's an example. The bottom picture I think is really good. On a prepped slide on the top, you can see a megakaryocyte, and you can see its long projection. When we talk sinusoid on the bottom picture, you get a better idea of what that is in your mind. It's within the bone marrow, and there will be some blood flow. So as the blood flows, again, it's going to take pieces of the roots of this cell with it. And within these fragments are all those chemicals that we briefly touched on. Hemostasis, not homeostasis, is the cessation of bleeding. So which is super relevant to make short, If we have hemostasis that's not controlled, okay, the relevance here is that you have blood volumes changing, blood pressure changing, that are going to disrupt general overall homeostasis. So there are 3 hemostatic, not homeostatic, mechanisms involved within blood control, blood clotting. That's the vascular spasm, the platelet plug formation, and then coagulation. Within the first picture, you can see that there's been some type of vessel injury where the blood wants to leave the vessel, or that would be a vessel, okay, or some type of tube or tissue. So with that, immediately what happens is it's almost like where the injury of the vessel occurs, there's a collapse. There's nothing to hold the wall up anymore. And then the platelets will be delivered via blood flow to the injury site and form the platelet plug. You can see on here as well that surrounding the vessel, that's a layer of collagen. Those collagen fibers are highly relevant as well. Around that platelet plug, we will have the actual coagulation or clot form via those collagen fibers kind of wrapping all around those platelets that got stuck at the end of the tube. Within the vascular spasm, this will prompt the constriction of the vessel, which gives us our first line of protection against that blood loss. Now, this can be caused by a pain receptor because some will directly innervate blood vessels in order to be able to do the constriction. Smooth muscle injury, and that has to do with the amount of smooth muscle within the vessels. And then the platelets that will release serotonin, actually, which is a vasoconstrictor. The effects of this will be that the vessel narrows, we stop some blood loss, and it will deliver some time. Within that platelet plug formation, it's temporary. Those we saw in that picture that the collagen fibers under the endothelium, were exposed. And then as the platelets congregated, they began to stick to the damaged vessel wall. The platelets will degranulate at this point. They will release a variety of substances. Serotonin, again, is one of our vasoconstrictors. ADP will attract and degranulate more platelets. There's a substance called thromboxane A2. This is for the promotion of platelet aggregation, degranulation, and vasoconstriction. This is a positive feedback cycle and will be active until there's a break or the break is sealed up. So then within the coagulation process, there is a conversion of fibrinogen. Remember, we heard that word. It's in plasma, not in serum, into something that's insoluble fibrin threads to form the netting that will capture the cells. Procoagulants or clotting factors, these are usually produced by the liver. We learned about a couple of them, and they're present in the plasma. They will activate one factor and it will activate the next to form a reaction cascade. Within this process, there are two pathways of how this happens then. There's one called an extrinsic pathway. This is the pathway of coagulation that stems from the release of damaged tissue. So the chemicals encapsulated within the tissue, when it becomes damaged, they get released and they will start coagulation via this extrinsic pathway. The intrinsic pathway involved in hemostasis and coagulation. These are the factors found in the blood. So like we just talked about how the platelet gets degranulated within the plasma, that in and of itself is what we classify as an intrinsic pathway. So adding a little bit more to that in this picture may help you. The extrinsic pathway, again, is initiated or catalyzed by the release of what is called tissue thromboplastin, or factor 3. This is from the damaged tissue. So if you bump yourself and you get a bruise, okay, this is happening. So the vessel gets damaged, tissue thromboplastin or factor 3 will be released in order to begin coagulation. Within the intrinsic pathway, this is done by the platelets themselves. The platelets themselves, these chunks of those megakaryocytes floating around in their blood, will release something called a Hogmann factor or factor 12. Again, this has to do with the coagulation process, the chemicals involved in it, and the pathways it may take. And it's both at one time. I mean, if you think about it, it's both things happening at one time. These coagulation cascades will lead to the formation of a substance that we call thrombin. We probably have heard that, and even if we haven't, we've heard of, we're doing thrombopoiesis. So that root word tells us we're doing something with platelets and coagulation. Thrombin that is released will then convert the fibrinogen, remember that, into the fibrin. This is a positive feedback mechanism. With the release of the thrombin, it's going to speed up the formation of what's called prothrombin activator and keep the clot going. So that's really nice. We have a positive feedback loop to make sure that the bleeding is stopped. However, what stops it? The fate of a blood clot will be stepped through via what's called clot retraction, and it takes about 30 minutes. The platelet-derived growth factor is secreted by the platelets and the endothelial cells. This is a mitotic stimulant for fibroblasts and also smooth muscle, so that there can be a repair of the damaged vessel. In fibrinolysis, okay, we saw fibrinogen that gets broken down into fibrin. The fibrinolysis, breaking down that, is what helps dissolve the clot. This is a conversion of plasminogen to a substance called plasmin, which is a fibrin dissolving enzyme. So let's go through some of the language again. Again, within the clotting phase of hemostasis, we have procoagulants or clotting factors that are going to catalyze and initiate a cascade of coagulation in one of two ways. It can go through an extrinsic pathway, which is done via chemicals within the tissue itself, or through the intrinsic pathway of coagulation formation through the platelets. Within the extrinsic pathway, there will be a release of what's called thromboplastin by the tissue or factor 3. In the intrinsic pathway, we're going to have the release of something called Hogmann factor or factor 12 done via platelets. So when these cascades get rolling, they will form then eventually thrombin. And if you look, I bumped the slide up, page 73, you can see on the bottom, okay, it pulls you out of the extrinsic mechanism or the intrinsic. They're both activated through tissue damage or whatever's happening on the inside. Eventually, okay, it gets down to the release of the thrombin. The thrombin is what will convert the fibrinogen into fibrin. And then this keeps going until we have something to shut the system down. Again, I'm not going to read through this entire chart, but I think will be helpful walking through how it's classified and broken down. The fate of blood clots, again, we walked through this. Let's look at some of the names of the chemicals. In the dissolving of the clot, we have something called platelet-derived growth factor that's going to be secreted by platelets and the tissue. So we have something from both pathways kind of helping to remove the clot. And then the fibrinolysis, breaking down the fibrin, is the actual dissolving of the clot when we have plasminogen converted into plasmin. And here is again, if If you don't like the words and you like the map, I would compare them, maybe put the slides side by side so you can see the difference. Something that's really important when we're talking about blood clotting as well is called platelet repulsion. We don't want the platelets to stick together and clot inappropriately, which we do see, some diseases and disorders result from this aberrant action. Platelets will not adhere to something called prostacyclin-coated endothelium or undamaged. Okay, so due to undamaged tissue not releasing or secreting any of those chemicals, the platelets won't want to stick. Thrombin dilution, this is done via rapidly flowing blood. Heart slowing and shock can result in clot formation because you can't push it. There are natural anticoagulants, heparin being one of them. It will interfere with the formation of prothrombin activator. And there's antithrombin from the liver, which will also deactivate thrombin. When we have a clotting disorder, one such disorder is hemophilia. This is a hereditary disorder because there will be a deficiency of one of the clotting factors. Physical exertion can cause bleeding and excruciating pain because there will be damage potentially to vessels. This is treated by the transfusion of plasma with purified clotting factors. Also, factor 8, which is produced by transgenic bacteria. Thrombosis, we hear this often, so let's do some language breakdown. This is abnormal clotting in an unbroken vessel. The thrombus is the actual clot, and it's most likely to occur in leg veins of inactive people. A pulmonary embolism, this is when a clot or thrombus breaks free and will travel from veins into the lung. When someone mentions they have an embolus, this is anything that can travel in the blood and block a vessel. Regarding infarction, this is tissue death, and this may occur if you have blockages to an organ like myocardial infarction or stroke. What we do clinically with blood clotting, the goal is to prevent the formation of clots or dissolve existing ones. In order to prevent them, we want vitamin K is required for the formation of clotting factors. So we might want to implement coumarin, warfarin, or Coumadin, and something that's a vitamin K antagonist. Aspirin also has this impact because it will suppress that thromboxane A2 that we mentioned previously. Okay, so there are other anticoagulants that we have used in the past. Medical leeches, medicinal leeches. If you have never heard about that, it's probably still done. It was done in convention into the middle of the last century. The application of leeches to a person to anticoagulate them via their released chemical called hirudin. Now, snake venom has an anticoagulator as well called arvin. That's the danger of a snake bite. So how do we dissolve a clot already formed? They have used clinically something called streptokinase. This is an enzyme made by strep bacteria. There's a lot of things that we utilize in treatment that really come from places you wouldn't think. One of them being strep bacteria in order to produce something for us in a therapeutic manner. There's something called TPA, or tissue plasminogen activator. This will work faster and is more specific. And now it is made by something called transgenic bacteria. And, you know, springboarding from the leech concept, something called Hementin, which is produced by an Amazon leech. So I love talking about things like that because I feel that we have somehow become removed from the natural state of things and we don't realize the origin of what we're doing. And I think that's really relevant and important. Anyway, that's it for the blood lecture. We'll see you in the next one.